Oh, I forgot to put the thing up. Oh, boy. From Microbe TV, this is TWiEVO, This Week in Evolution, episode number 90, recorded on May 10th, 2023. I'm Vincent Racaniello, and you're listening to the podcast on the biology behind what makes us tick. Joining me today from Salt Lake City, Nels LD.
Hey, Vincent. Good to be with you. Greetings from LD Lab Studios. Speaking of studios, I was just almost in range of you there at the incubator last weekend. I was at a meeting on Roosevelt Island, bordered by the East River. This is the Jane Coffin Childs Foundation, outfit that sponsors postdoc fellowships for 3-year runs and has an annual meeting, just coming back into person with the pandemic for the first time. So we were out there. I'm on the Science Board of Advisors, and we were out there to pick next year's postdoc class. Stay tuned for almost 30 really incredible up-and-coming scientists. And then the current ones were there to give their updates on the work they've been doing the last few years. And so, yeah, and I was just, kind of wishing I had just a few more hours or a day to potentially swing by your place to go in vivo, whereas this is still in the works. Didn't quite pull it off with the timing this time, but there was a virology connection. Are you familiar with this sort of the ruins of a virology hospital, a smallpox hospital on Roosevelt Island? Have you been there?
Well, I've gone by in a boat, right? Because I'm not sure you actually visit the smallpox hospital. It's fenced off and it's in ruins, right? It's all falling apart, but I've seen it. It's pretty cool. Yeah.
Yeah. And actually recommendation to step foot on the island. You have to take the tram, or you can take the tram, I should say, from, right in the shadow of the Queensboro Bridge, or
correct.
You can take the F train and get over. There's a stop on the subway as well. And pretty great. Actually, it's a neat place to have a meeting. So the Cornell Technical College, part of Cornell University, has just put in place a couple of massive buildings as part of some of their master's programs, I think, in technology and some of the applied stuff, technical stuff. And then the graduate hotel chain has a pretty big footprint there, big hotel with a great rooftop bar, incidentally, the Panorama Room, worth checking out, I would say, under any occasion. Yeah.
So people who live there tell me it's boring because there's nothing to do.
Yeah, I can see that. I mean, it's really wild, right? I mean, you're literally just looking across the river, the East River, to part of Rockefeller's new campus.
Right.
It's like Midtown Manhattan, I guess. Like one of the stops is like 57th and Lex. Just you're not too far from the heart of the action, and yet somehow even just that East River is quite a barrier, both sort of aquatically and maybe psychologically somehow. Yeah, pretty wild. But
one of my TAs for virology this year, he said he used to live on Roosevelt Island, but they got bored and because there's nothing to do. It's just apartments, and I guess this hotel is new. But why would you go to a hotel anyway if you're living there? So they moved back into Manhattan, he and his girlfriend, because nothing to do there.
Fair enough. I could see that. Although hopping on the subway, I mean, the activation energy for getting to Central Park is you're there in 20 minutes.
It's not a big deal. Yeah. I mean, if you live in Queens or Brooklyn, there's stuff to do there if you don't feel like going into Manhattan, right? But the problem with Roosevelt Island is there's nothing to do. So
that's right.
But I've never actually been to the island. I've never been on it. I've just gone by it in boats. I've seen it from Manhattan, and so forth.
So
that's cool.
Pretty wild.
Next time, try and get by. That would be cool.
We'll do it. Yeah, it'll happen again.
So welcome, everybody, to another Wednesday afternoon TWiEVO. Get your fill of evolution. I want to thank our moderators for coming by. Barb Mack is here. Les is here. I also have seen Andrew from New Zealand. Of course, Barb Mack's from the UK. Les is from California. Andrew's from New Zealand. And I also saw Tom here as well. Tom
Fantastic.
he's in Oregon at the moment.
Mm-hmm.
And those are our moderators today. Steph, I see Steph has just shown up. Welcome. Steph is from San Francisco, as reflected in her name, Steph SF. Let's see where people are from, Nels. What do you think?
Yeah. Let's go around the thread and see
All
where
right.
we
We
are.
got Claire from the UK. By the
Welcome.
way, where is Lise? We'll get to her. Okay. Claire is from the UK. Nicola is from Italy. I know he put that up earlier. Let's see. Who else? Philip is also from the UK, Wales. There's Tom from Eugene. Lori's from Tomball, Texas. Right. Now, Lise is from Columbus. Lise visited the incubator on Monday.
Oh, very cool.
She was in New York for the weekend. She stopped by, so good to see you there. That was pretty cool. Yeah. What else do we have here? Let's see. I have to scroll down. Susan is from upstate New York, not too far from where I am. That's good.
Yeah, welcome.
And we have Noir from New Mexico. Good to see you again. Noir was at the 1000th celebration of TWiEVO.
Very cool.
Peter's from Boulder, Colorado. Peter is from Stockholm. I love this stuff. I just love it.
I do too.
Maureen is from northeastern Ohio. Elcio is from Brazil. We have someone from New York. Hey, Kip. Good to see you again. And we have about 40 people here today, which is really cool that we have anybody here in the middle of a Wednesday afternoon.
I agree. Well, depending where you are in the world, it's a different time of day. But what a great.
Jamie's from Wisconsin.
Welcome, Jamie. What a great crew from all over. Yeah.
And Rob has just given us our first donation. Thank you, Rob, for your support of science education.
That's awesome.
Tonight, by the way, tonight on our live stream, Office Hours, I have a special guest, Dan Wilson from Debunk the Funk, which is a YouTube channel. He's been on TWiEVO before, but I was recording a video with him, and I said, why don't you come on our Office Hours? So he's going to do that. Look at this. Irene is from Cyprus.
Wow,
cool.
Welcome. Yeah.
Cyprus is an island in the Mediterranean, right?
Yeah, that's right.
Have you ever been?
I haven't. Have you?
Nope.
Add that to the list. Yeah. Same
I don't know if I don't think I'm going to get everywhere in the world. It's just too many places. But all right, folks, thanks for coming. Let's dive into some science, Nels. What's on the plate for today?
Yeah, I agree. So we've assembled a crew of curious, creative people, which is the perfect time to dive into some science and kick around some ideas. Everyone out there, as questions are coming up or your ideas, get those into the thread, and Vincent will be lifting some of those up so that we can respond directly and in real time. And we're back a little bit quickly here, Vincent. I think it's only been 3 weeks, but who's counting, as the old saying goes. And it's always good to be together discussing science. And I know with your upcoming travel schedule, that's kind of why we decided to sneak this one in a little bit early, make it a little bit easy on the two of us. So we've got, I think, a pretty interesting paper here today. And honestly, I have to tell you, actually, just in the last 3 weeks, Vincent, there's been sort of a burst of pretty cool publications, either bioarchive manuscripts. We'll see one today. Some all the way to the sort of fancy glossy magazines of science, showcasing a lot of interesting evolutionary studies. And so we might touch on a few of those at the end here. It was actually pretty tricky to pick just one paper today. And so I'll confess to you and our live streaming crew here that I pressed a little bit of the easy button. I picked a paper that is, it's on the bioarchive. So first of all, it's actually freely accessible, which is important, open access to science, so that we can all dive in and look at the entire presentation, all of the details for ourselves, and to go deeper to sort of, follow that curiosity, follow our noses. And if something's behind a paywall, that's a lot harder to do. So this one's on the bioarchive, and it's also, we'll see, I don't think we're going to be spending a lot of our time like really digging in deeply to the sort of the concepts or walking through them. It's a pretty simple finding, a pretty simple presentation, but I think it has some really cool sort of sparks for thinking about some evolutionary concepts and what's sort of possible by considering or discovering what's going on in the world around us. So the paper, or the preprint, I should say, is entitled Genomic and Radiocarbon Insights into the Mystery of Mouse Mummies on the Summits of Greater than 6,000 Meter Andean Volcanoes. And so, so we're not in Brazil at the moment, but we are, as one of our live streamers is, but we're not too far away. We're in South America, and we're in Chile, and among the ranges of the Andean mountains. And we'll talk about some of those details, the extreme environments here, and these mysterious mouse mummies, and how they got there, and what that might mean for thinking about sort of the evolution to extreme environments, or even the evolutionary capacity potentially for mammals in this case to live in these high elevation extreme settings. But before we get there, let's take a moment to lift up the authors here. So it's first author Jay Stortz. He's in the School of Biological Sciences at the University of Nebraska-Lincoln. Very different elevation from the Andes volcanoes, to say the least, in the upper Midwest there. And then group of folks who are located in in Chile, in Valdivia, Chile, in Institute of Evolution there at the University of Austral de Chile. And so this is Marcelia Quirona Carmona. This is Juan Opazo, Guillermo Dialfea, and from that crowd. And then there's another collaborator here, the last author, Jeff Good, who's also a slightly more mountainous region in the U.S., but this is the School of Biological Sciences, or Division of Biological Sciences, University of Montana in Missoula. And so give Jeff a little credit for some elevation compared to Jay. But the real game is in Chile, as this sort of multinational team that came together to do the work is showcasing. So that's the group. I would say, a little bit of background. So actually I know Jeff Good pretty well. He's a really interesting scientist up in evolutionary biologist, population geneticist up in Montana. Some really cool work on sort of mammalian adaptations to environment. So like the snow hares, these rabbits that have different fur colors in the summer versus the winter for camouflage. They study some of the, sort of mechanistic basis of that, the evolutionary, or, both the mechanics of how that works at the molecular level, and then how that evolved in the first place, and various topics like that. Jay Stortz does a lot on the evolution of sort of metabolism, getting into the biochemistry even. And I think, this work that we're discussing today and the work they present will start to open the door to some of those questions. I think about, how do you pull off the metabolism of being at around 6,000 meters, in the neighborhood of 20,000 feet elevation. And so I think some of the work here lays the groundwork for thinking about that, and starting to get these mice potentially even into the laboratory. But let's deal with the work in front of us here instead of speculating about the future, and then we can do that at the end. So I think one of the big questions here that's pretty interesting to me, and hopefully for other TWiEVO listeners and TWiEVO enthusiasts, is the notion of evolution of life in extreme environments. And
New York. Nels.
That's right, absolutely. You can like the pizza, whether it's the pizza rat or a mouse living on a 20,000 foot volcano. There's a lot of extreme environments out there. Maybe echoing on what you just said about how hard it is to think about traveling the world. Now add all of the complexity around us, or if we go into the oceans, and these elephant seals that can swim almost 3,000 feet deep, and what does it take to do that? So there have been all of these great, almost natural experiments in genetic adaptation, or the evolution of the ability to tolerate, or even in some cases thrive in these extreme environments. And of course, the sort of payoff for this from a kind of biological standpoint is that if you can if you can live, if you can operate, if you can reproduce in an extreme environment where other species can't, that might unlock the resources or the space to sort of either gain more resources without competition or avoid things like predators, and to have the space to be safe. That'll be, I think, an open question actually as we step through this piece, and with some we might solve the mystery of the mouse mummy to some degree, but it will open up, as good science always does, it will open up some other mysteries along the way. So the species here is the Andean leaf-eared mouse found in the Andes Mountains, and I just dropped a photo of one of these guys. Is it possible, Vincent, for you to screen? There we go. Yeah. So there it is. I think you can see a little bit how it got its name. That
It's
pretty
cute.
big. Yeah, cute mouse there. I think that photo is actually taken from an earlier study by some members of the same group, from the Storts group, where they were, I think, back in the 2020, about 3 years ago, were able to actually live capture this example here, and at the time it broke the record for highest living mammal to be located, with the assumption that it's living there. And actually, this is perfect. The way you've set this up, Vincent, on the screen, we've got the quote here from the intro of the paper. So I'll just recite that. So on the summits of these 6,000 plus meter Andean volcanoes, plant life is non-existent. Temperatures less than 30 degrees C are common even in the summertime. Wind speeds can reach 200 kilometers an hour, and there's less than half the oxygen available than there is at sea level. For such reasons, these extreme elevations were previously thought to be completely uninhabitable by mammals. And so, giving lie to that uninhabitability is that critter that we see in front of us now. This is the Andean leaf-eared mouse. And so, some live captures, but as they were doing that, they were able to, and by the way, they were sampling, I think, at least 3 volcanoes, Andean volcanoes, here in Chile. And we're not only recovering some live mice from that. I think they got about 40 or so, not a ton, but they're just sampling. They're not surveying the whole population here. And it's worth it if all of you out there, if you haven't already, to just go on the bioarchive. We'll have the links are up and around. Or if you do it, you should be able to find it, and spend a minute with the photos of just how uninhabitable these locations look. I mean, these are just like desert-like volcanoes.
Let's
maybe
put
with
a
a little
couple
bit of
of,
snow.
let me grab it. There's some in the
Okay.
paper here. Right?
Yeah, exactly. Yeah, you can just put the figure up.
Let me
And
put this up. Yeah.
yeah, you can actually see
go.
these volcanoes. Yeah, here we go. So this gives us the map of where we're located.
Yeah.
And you can really see, like, if you're if you're down at the bottom of one of these volcanoes, can you imagine just like, what are you thinking when you want to climb to the top of that thing and live up there? Yeah. So here's the 3
pretty
volcanoes.
desolate, right?
Yeah, it's pretty to say the least. That's being generous. Extremely, extremely desolate. And there you see one of the mummies. So this is, that was one of those, that cute furry guy. But they were able to recover, I think, about a dozen or so of those little sort of, mouse popsicles in a sense, and actually, as much as it's cold and there's snow up there, the wind is howling. It's also, maybe one of the biggest features of the extreme environment is how dry it is. So this is like a massively desiccating environment. Actually, as we'll see in a few minutes, that can have some big advantages for preserving the DNA of that so-called mummy. Obviously not wrapped in any way here, but that actually worked in their favor to actually be able to recover the DNA to do some real high quality genomics moving forward. And so, the question they set up here actually is, given that it's so extreme, as we've been sort of glimpsing at here, is the question of whether, how did those mice get there in the first place? And so are they living up there? Are they reproducing up there? Which I think is still an open question. Or, were they brought there by humans, by our own species, who, in particular history, comes into play here with the Incan societies and some of the rituals, including including human sacrifices in some cases, and also bringing other offerings basically to the gods, which could include these mice. And so that was one of the questions that they wanted to get at, was the motivation for doing radiocarbon dating was to see if there's a match or difference in the timing of when those mummies became mummies. And then also to do the DNA sequencing. So there's you can compare the population genetics sort of components to determine whether the patterns you see in these samples that you sequence match local populations, which are at lower elevation, but nearby the volcanoes, or were potentially transported from farther afield. And that again would be potentially more consistent, that they were transported as part of some of human activity tracing back a few thousand years. Okay, so in the end, 13 from the sampling, 13 mummies, 3 volcanoes. And immediately the radiocarbon dating sort of starts to provide evidence that's not consistent.
Hold on.
Yeah.
Can I interject?
Yeah, of course.
I just want to read a quote. So the introduction to the paper, they have a quote from Ernest Hemingway.
Oh, that's right. Yeah, that's a great one. Yep.
It's from the snows of Kilimanjaro. Kilimanjaro is a snow-covered mountain, 19,710 feet high, is said to be the highest mountain in Africa. Close to its summit there is the dried and frozen carcass of a leopard. No one has explained what the leopard was seeking at that altitude. I just think, a science paper, I hope they let them leave it in. I think that's great, because this is what we're looking at in this paper too, Nels.
That's right. Yeah, they kind of drew inspiration from Hemingway to then motivate the study here. I love that. I agree. We'll see what happens. Actually, that'll be interesting to watch this paper as it steps through. So, for example, I think that photo of the mouse, we were just gazing at the live one from a couple of years ago. That paper, just that first report, even that sort of record-breaking mammals at 6,000 meters, ended up published as sort of a short note in PNAS, the Proceedings of the National Academy of Sciences. It'll be interesting to see sort of where this one graduates. And like you're saying, does the quote make it in to sort of add that sort of inspiration as well? Okay, so back to the radiocarbon dating though. So all of these samples, there's a range of times, but like basically in and around 500 years ago, plus or minus the confidence intervals, which can be pretty wide with this method of radiocarbon dating, but still puts it pretty far outside the range of at least the specific hypothesis that the Incas were bringing the mice with them as they were exploring, exercising sort of cultural rituals up at the tops of these volcanoes. And so that already is saying, and by the way, there's also, right, the fact that you have a picture of a live mouse there and they're trapping live ones is pretty consistent with the mice are, pretty regularly getting there on their own. But that's still, sort of leaves open a lot of the questions about their getting there. But are they living there? Are they reproducing there? Are they in enough numbers? And so maybe a little hint of some circumstantial evidence that there's, that they might find a mummy around like a pretty full specimen like the one we just saw from Figure 1. But then there's like just sort of bones and partial remains that might have been, less preserved or gotten sort of battered or bashed around and didn't do as well. And actually from those remains, they're also able to, without, looking at the DNA, make a pretty clean identification to the genus of mice, Phyllotus. And so, and already there's some, not genomes, but mitochondrial DNA evidence or mitochondrial DNA data that helps to actually, make that sort of molecular level DNA-based genetic connection between the species that they're dealing with there. And I guess as, petrified mummies, not so clear like what that thing would look like. And so the skeleton helps a little bit to place it there, but then the real sort of link is in the DNA. And so, a couple things here. So one is, I think the closest actually full genome is the hispid cotton rat. And they only share a common ancestor in that the genus of these Phyllotus mice 10 million years ago. And so, just sort of a side note here, but it really illustrates how kind of, sparse our sampling is, even of mammals. And in part because these a lot of these critters are living in these extreme environments or in places where there's not a lot of humans. And there's so much genetic diversity that we're just, basically in our infancy of understanding the genetics of all of the diversity of life around us. And so just, that in some ways it feels like we're kind of in the post-genome era. We sequence genomes and, we must know what everything looks like. And in fact, we're massively undersampled in a way. The good news is there's all this great, increasingly powerful technology that's like almost out of the box DIY genomics that is possible. And so here they could get chromosome level de novo assembly. So even though no one had really sequenced a close relative or even a species in the genus of these mice, just by getting some good DNA, which was like an assist on the extreme desiccating environment there to preserve the DNA for hundreds of years with like basically no signs of sort of the mutations that are common from ancient samples as the double strands degrade and become mutated and sort of littered with problems. This also helps, like, because they've preserved the DNA so long, it also helps with the contamination problem. You can sort of go after the mouse DNA, and the samples are like the vast majority of it is exactly the target that you're aiming for. And so between both long read sequences, so they use the PacBio technology, and short read, the Illumina, they could very quickly get to basically they picked one mouse and got to 30x coverage. And by the way, that mouse they picked isn't one of these sort of like, super extreme, like ultra marathoners to the top of the mountain. This is a live trapped female from about 5,000 meters. We're still talking, on the order of 15,000 feet here. This is still a pretty high environment, but not that sort of like the extra 1,000 meters. A lot happens in terms of the environment. And then, maybe Vincent, a little bit of an echo from our recent consideration of a different kind of extreme environment, the dogs around Chernobyl. Remember, we picked up that paper a couple episodes ago on TWiEVO. So there's real value as evolutionary biologists, evolutionary geneticists, population geneticists, to not just sequencing one, but sequencing many. And in fact, sequencing close relatives in these cases, because now you really start to get the genetic context of who's related to who, what kind of diversity, and so, with that idea that you potentially could be getting a sense of, and through those comparisons, what does adaptation look like as you push that last 1,000 meters, or consider, the closest relatives that live at lower elevations compared to the ones that live higher. So they don't actually do that analysis here, but I think they're setting themselves up by taking 40 other samples, including those 13 mummies, which adds another interesting comparison point, because these are now 500 years old or some range. So now you've turned back the clock. You have sort of the diversity of today, but then you can compare that to the diversity from 500 years ago, or sort of that gradient of times, and ask, some really interesting and specific questions about how these things might be evolving almost right before our eyes, or over the course of a few centuries or something like that. And so, I wouldn't be surprised if there's more sampling going on if to sort of build out that gradient in time and space, where time being these ancient mummified samples and space being as you traverse up to the top of these volcanoes. So roughly equal male and female, I think they threw in that detail to at least, kind of a circumstantial point that there are enough potentially breeding populations that could be sort of making it up there and, sort of eking out an existence for like generations to come. If that's true, now, sort of that power of mutation and selection and the recombination happening with F1s, F2s, F3s, you could imagine that, that could unlock maybe some mutational situations that could actually, where natural selection could act to then fortify that population.
Yeah, they say that typically dispersal in small rodents is sex biased and happens over short distances. But so the fact they have equal males and females in both the mummies and the live captured, right? It says they're probably self-sustaining populations that have been there. They're not just coming through, walking through and seeing what's up.
That's right. That's the idea. And I think, that's some, at least circumstantial evidence to support that. I don't know if they've really proven that. And to me, that's almost even more interesting. So, evolution is mostly things that we don't see, right? So these are
Yeah,
like
yeah,
all the countless number of mutations that sort of fail, or the attempts at something. You try to adapt to a new environment, or
yeah.
a pandemic virus sweeps through your population, and sort of the ones who live or go on are very different. And so, like, to me, what was really fun about, or kind of piqued my curiosity about when reading this preprint, was that idea that maybe there's things like the parts of a failed experiment, like, so maybe that's the mystery of the mummies. They all become mummies because they go up there, um, but in a big enough population that something could be possible, but they're not quite there yet, right? They all become mummies. And so, but that, I think that's the mystery that will probably persist, or that they're continuing to work on as science moves forward here, is whether that live trapping that they're doing and are successful at, is that from a reservoir of local residents, or is there some ongoing experiment here where the lower elevation ones are motivated for who knows why. And I
Mm.
think they sort of bring that up at the end. But they're making the attempt, they're doing the experiment, the natural experiment in real time. And whether that's still a failed experiment or if it's succeeding, I think is an interesting question.
Mm.
In the environment itself, that's not a unchanging thing either. So obviously, as climate change becomes more impactful, um, being first to 6,000 meters and to be able to sustain or set up a breeding colony there, then imagine, as that environment might get less extreme by some variables, more hospitable, maybe food will show up. Remember, there's no plant life here. What are so if they are living up there, what are they eating?
Eating.
Where,
That's the
where
thing. That's
yeah,
the big question for me because
yeah.
you can't have a sustainable community without food. There's no plants, right? And that's what they would probably eat, right?
Correct. Yeah, exactly. I believe that's the diet of these things. That's exactly right. If there's 200-kilometer-an-hour winds, I don't think there's going to be bugs moving through. I mean, there's just so the amount of calories
Yeah.
available there. So certainly you've solved the problem of predators if there's no other life up there, but you've
Yeah.
created a new one,
You
which is
starve.
how do you? Yeah, yeah.
But Nels, how far would they come from? Like, what's the level? What is the elevation where there would be more hospitable conditions, where there'd be plant life? Do you have any
Yeah.
sense for that?
Not much, but I think it's a great question, and I think that it's not that far, honestly. So, the fact that the fact that they use a mouse for the genomics, which is a live trapped individual from 5,000 meters, I think even as you're,
Hmm.
just go down that elevation. I mean, you can see from those photos we looked at that it's a pretty you have to go down quite a ways, but that does raise that question about migrations, or or even um, sort of ranges. So is that population, are these
Yeah.
like mountain climbers that go up to the top of Everest but then come down, or they restock each other? Are there mouse Sherpas here that are they transporting? Are they getting that foothold? There's really fascinating questions that I think they're just they're setting up the system potentially to get there. Um, and we're just getting sort of that first glimpse from the very top that sort of outlines how sort of challenging or daunting it might be to live there. So, and
So,
then, yeah,
so Tom points out. Okay, here, mice often store food. I wonder if over time the population developed higher and higher caches to avoid
yeah,
competition and
yeah.
predation. So they could have brought up some food with them, right? Although I don't know how they'd
Mhm.
know to do that, right? Because if you went up without food and you died, that nobody would know who you
Who's
left
gonna
behind.
who's gonna remember? Yeah. Well,
And
I like this. Yeah,
he
yeah,
also
yeah.
asked whether lichens would be
Mm,
up there, because there should be some lichens because
yeah.
they're stuck and they're not going to go. They're not going to get blown away, right?
Yeah, absolutely. And so I love, these are great points, and raises sort of that potential pathway forward, and I think in two ways. So one is, and to your point too, Vincent. So, this idea of, if you are stashing food or caching food at different locations, but you don't survive there to reproduce. But if that's an innate behavior of your species, then it's sort of, that's cooked in. And then when it's mixed with this other behavior, which is this sort of like motivation to explore higher, or to get away from predators, to
Mm.
get away from competition, then it's the possibilities that the combination of those innate behaviors sort of work in your favor, even if you don't necessarily reproduce or make it. With enough time, you could start to see that sort of gradual kind of that evolutionary foothold in a sense, where maybe these things would start to work together. And I also really like the point about lichens, right? Or so not just caching food, but in some cases bringing it with. And so the first sort of one obvious example that comes to mind are some of the complexity that you see even among insects, right? So like leaf cutting ants, which will aren't eating the leaves, they're cutting them, bringing them back to their colony and cultivating fungus on them. And so, yeah, I wouldn't put it past, mice or other mammals to also, have some of these more complex strategies where these mice could start to become high elevation farmers in a sense, where as they're bringing seeds and caching them, that's also an experiment for the early colonization steps in adaptation of plants, lichens, other life that might be just sort of barely able to hold on to these extreme environments as well.
So,
And
Nels,
so,
I
yeah.
looked up how far a mouse can go in a day. So,
Uh-oh.
a mouse can go about 4 kilometers a day.
Mm-hmm.
And of course, that's going to depend on the species. These guys may be hardier. And this 6,000 meters is 6 kilometers, right? So they could get there in 2 days, really. So, if they're walking continuously, that is.
That's right. Well, and I think what you described is like a complete vertical descent. So, thankfully, hopefully there's, you're doing this over sort of longer distances so that you don't have to like also be able to sort of have like a Spider Man like quality of being able to cling to the side of a building. But yeah, no, that's right. So there's no question. There's like a lot of capacity here. In these rodents, they're pretty robust, pretty strong. They're already living in pretty extreme environments to begin with. Absolutely. So then, so I think that's kind of one of the fundamental questions that we're sort of talking about here is basically, so, how far along in this process are these mouse populations? And so if you start to do the population genetics, you can actually maybe, sort of get some inferences about like, have they been living here for a really long time? Are these new explorations? And so the way to get at that is, to ask about the genetic diversity between the samples that you have and how that relates to the other ones in the population. We spent a lot of time on that a couple of TWiEVOs ago, talking about the dogs and of Chernobyl and who do those dog populations, who do they most resemble genetically? So they do that here. It's a smaller sample. It's the 40 or so. And they do some similar principal components analysis to try to get a sense of like, how do these things hold together in terms of like all of the shared or differentiated genetic sort of diversity here. And unlike the Chernobyl dogs and the different breeds and that probably history and the differentiation of dog breeds even, here, all of the sampling, between the different volcanoes, by the way, which are geographically, you can see in the maps how these are geographically distinct. The mice are all kind of, sort of in similar space. So an alternate outcome could have been if you're on volcano 1, the green triangles, that's one sort of set of structural differentiation of the genomes or mutational shared differences. You could have, and then the other 3 volcanoes, those move in different directions. In this case, like it's somewhat limited sampling, but they're all kind of falling together. And so, I think what that implies is that if this is sort of the beginnings of adapting to this high environment in new ways, it hasn't really happened yet. Like they haven't sort of, I think what you would expect if this was like a long, successful, ongoing process is that as they got to the different volcanoes, they would sort of become their own distinct genetic populations as there's a lot of inbreeding and sort of the development of the distinct populations. And you don't see that. And I think that's pretty consistent with these are still like early evolutionary forays. And it's almost like, again, perhaps that natural experiment is just getting underway. If you come back in 10,000 years, 50,000 years, 100,000 years, then maybe like that story will have already been kind of litigated by mutation and selection.
Yeah, it is. It is what the oldest mummies are 500 years, right?
Oh, aren't they get a lot older, don't they? Oh, sorry, these mummies, the mouse mummies. Yeah, no, right. Yeah, that's right. Yeah. Yep.
Of course, that's nothing in in evolutionary time right now.
Oh no, yeah, that's right.
Unless you're a
Like,
virus. Unless you're a virus.
yeah, although well, a lot can happen in 500 years is pretty surprising, but yeah, in the big picture here, and I think a complex sort of establishing a new niche where the environment is this extreme, it's really a pretty unique challenge that hasn't been conquered. And I think the authors are willing to sort of throw up their hands as well. So another quote from later in the preprint: "We have no explanation for what the animals were seeking at those altitudes. Mice climb to the icy, wind-scoured summits of greater than 6,000 meter Andean volcanoes for reasons that remain mysterious." And so they're not willing to kind of go on the record with a sort of really speculative sort of situation here.
And what would you think they need to do to really understand what's going on here, Nels, assuming they could do more sampling?
Yeah, well, I think something that will be probably really fun, and I hope is in the works, is they've been they didn't sort of sacrifice all of their samples to do the genomes. So if I'm curious, I don't know the answer, if it's possible at lower elevations, say in Lincoln, Nebraska, or maybe a little higher elevation in Missoula, Montana, to set up breeding colonies of the leaf-eared mice. If that's true, then you can start to put them through their paces, right? So like at high and low elevation, does this change their metabolism? How do they compare to like common house mice that live at lower elevations, or some of the closer relatives that might be at lower elevations? And can you start to put like, genetic changes into place here? And then the most interesting question is almost feels to me like the biology of the future here. So if they do start to like gain more of a foothold and live up there, like what does that look like? I mean, you could imagine even like sort of as experimentalists sort of stacking the deck a little bit, and like so doing what it looks like to get these experiments approved, but to set up mouse cages at various parts on the Andean volcano range and at various elevations. And then you might like provide them with a resource, food, on a regular basis, and sort of take away that, imperative, which might like have like a massive, strong selective pressure against establishing a colony. But then the question could become really interesting is like, over 5 years, 10 years, what does breeding look like at 6,000 meters versus 5,500 meters, 5,000 meters? And you could really get a sense of like, you could start to control some variables and get a sense at sort of like, what is the sort of potential of the adaptive process as you add and take away sort of various key variables. The closest study I can think of that comes to mind sort of along these lines are there's some really cool, these might also be in South America, some really cool experiments with guppies. And so, and you can just go to your pet store and see these like super bright, colorful, live-bearing fish. The males just have these spectacular patterns and colors. And so some evolutionary biologists set up some ponds where, so out in the field, set up some ponds, but they did the thing that they controlled were the predators. So in pond 1, it has predators. Pond 2 doesn't. And then you have replicates, et cetera. Let the populations of guppies hang out and breed over the course of several generations, and then come back and ask, are there like, obvious or noticeable differences? And the answer is yes. So in the predator stocked ponds with the male guppies become more drab. They lose their sort of their bright colors. Whereas you go to the, either without predators or maybe even more dramatically, when you put these things into pet stores and then breeders like, vie over who's the coolest, weirdest looking guppy, then you're selecting really strongly for the most fancy, wild, colorations that might grab our own eye. And so, these are, fish are not mammals, but they're, share some of the same experimentally favorable features, which are short generation times, the ability to like house or create large populations and those kind of things. And so, I think the other thing that got me about this preprint is it sort of has that sort of, the ingredients for that cool, like going out to nature, seeing all the complexity around you, but also the ability to start to bring some evolution or some experimental ideas to the mix, and And this preprint, I think, lays the groundwork by doing the genomics.
So let's do some thinking here. So,
Yeah.
you were suggesting these experiments where you, in the U.S. at different altitudes, you establish breeding colonies, and I was thinking, well, why don't we just go to 6,000 meters on these volcanoes and
Oh,
put
yeah.
a bunch of mice, and then come back and check them every week or whatever. But that's probably not how it happened. It probably the mice gradually went up higher and higher, right? And maybe they bred for a year or more at each level, and then until they moved on, right? So it was probably a gradual thing. So it may not be that you can throw mice up there and expect that they're going to survive, right?
Absolutely right. Yeah, you might just make a bunch more mummies, right? And so
Yes.
it'll be the mouse mummy creation project or something like that.
And also
No,
remember, we have to consider the possibility that they brought food with them as they were. I
yeah,
don't know
yeah,
that. Do mice do that? I have no idea. I
yeah,
just
they
don't
do.
know.
They do, and that was Tom's point, sort of that behavior of caching food, and so
Yeah.
and they might even, they may never get back to it, even under sort of like the best of circumstances or the worst as well. But no, your point, I love that point, Vincent, because it's like it's sort of that tension between observing nature and evolutionary processes unfolding, and sort of the big compromise there is it takes a really long time, depending on what you're trying to
Yeah.
sort of study. Um, Versus, and then but then all of the favorable things about, like, it's natural, it's real life, it's the quote unquote natural experiment. But then that's the question: as experimentalists, as evolutionary biologists, but also experimentalists, can we go in there and make necessary compromises where it's not the real process? But if we make the choice, like interesting choices about how we set up the experiment, it might give us, some of the features of how this evolution is happening that might not completely recreate it, but it might like speed up the process or reveal some of the rules going forward. And so I think that's, you've put your thumb on one of the challenges and opportunities of being an experimental evolutionist, is that you're constantly trying to figure out, how do I design an experiment that maximizes what I can learn about this, and minimizes the compromises to the real system, which is, you can't do all of the complexity. You can't sort of replay the tape of evolution over the course of, like, our lifetimes, or maybe even a scientific career, or let alone the course of a 5-year funded grant, right? I mean, so
Yeah.
like that
Yeah.
puts a lot of other pressure sort of on how you think about some of these approaches.
Well,
Yeah.
what I would do is see at what level, the highest level, you find consistent mouse populations on these slopes,
Mm-hmm.
right?
Mm-hmm.
And then pick something intermediate to the summit and try and establish a colony. You could capture some of those animals and bring them up. I don't know. You could do 1,000 meters at a time and leave them there for X amount of time. Check them, see if they are breeding or if they're all eaten by predators or whatever. I mean, it obviously takes a concerted effort, funding, and people willing to spend time, because I think you have to stay there on the slopes probably.
Oh, yeah, exactly. No, and you raised another, like, really interesting point here, that I was going to bring up too, which is, so this kind of this direction reminds me of some pretty cool work that's already happening, not up in on high elevation volcanoes, but actually in the woods around Princeton University. So some
Mm.
colleagues, Andrea Graham, who's a great kind of ecology, evolution of infectious disease scientist, Ken Cadwell, who's a really good mouse model systems of sort of complicated infection biology. They've been pursuing these, what they're calling mouse rewilding experiments. So this isn't the Andean leaf-eared mouse that we've been talking about, but Mus musculus, right? And not even just like Mus musculus, but the inbred strains that
Mm.
immunologists, other geneticists have been using now for decades. And so the idea is to take these lab mice and put them back in the wild, and then see what that means in terms of their
Mm.
susceptibility to
Mhm.
virus infections, bacterial infections, et cetera. And so the reason I bring it up is, like, your point about, like, being there all the time. So getting Ayakuk to approve a mouse protocol where you
Yeah.
put these, so in this case, they've built these sort of pie, like round enclosures with little dividers. It's almost like cutting a pie into segments. And then it's, like literally in the woods. And And so Ayakuk, immediately approaches them and says, hold on, we're really nervous that you're just putting these mice out in the wild.
Yeah.
What if they're,
Yeah.
what if the bears come and eat them or something like that? And
Sure.
that's right. Yeah. It gets worse. So then the solution from Ayakuk is that there should be like, undergrads and graduate students camping out 24/7. Like the first
Yeah.
line for the bears should be the graduate students so that the mice are safe. I mean, it's that level of like red tape to start to think about doing even some simple experiments like
So,
this.
so what Nels is
Yeah.
referring to, IACUC, is the Institutional Animal
Mm,
Care and Use Committee. If you want to do experiments
that's right.
at your in your lab, you have to follow. You file a protocol and it gets looked at, and they make sure that you're doing everything according to the rules. And one of them is if you're subjecting them to any kind of stress, you have
Yeah.
to check them periodically to make sure they don't have undue suffering. Right. So
Mm,
when we used to infect mice with polio virus, we had to check them every 12 hours because as soon as they get paralyzed, you have to euthanize them. And so
yeah.
I could imagine the mice in the in you put them up in there, and they say, "Oh, you have to check them every 12 hours." Make sure
Yeah.
so you'd have to live up there.
Exactly.
Yeah,
No, yeah, that's right. Yeah. Or even worse, you have to protect them. I guess for the Andean volcanoes, the good news is there's no bears up there. It's such an extreme environment that
yeah.
you've solved the predator problem. But yeah, no, the logistics of doing a research study at 6,000 meters, 5,000 meters, I think is not insignificant, but pretty interesting. Yeah. And so
I
I'm really
mean, just
curious. Yep.
just
Go ahead.
water. Where are they going to get water from? Right at that elevation, there's no pools of standing water. I don't know what they would do. Right.
Yeah, no. So I think you're seeing from those photos, like there's snow, and so what the freeze
Okay.
thaw cycle is, and the strength of the sun and various things like that. There is even there, yeah, there was someone who I ran into from the University of Washington at another meeting not too long ago, and he actually is a neuroscientist, but he's also like an avid backcountry skier, and so he was like found there's these snow flies that you can see up there
Hmm.
in like literally like living on snow and somehow reproducing. These are insects, and so just from his backcountry skiing habit, he got really into like collecting these snowflies and trying to figure out have they adapted to that sort of extreme environment. And actually the answer in that case is probably no. Like they don't have antifreeze proteins or things like this. They're just sort of like have figured out how to subsist um, and get away with sort of surviving in cold environments. Here it's like much less extreme. We're just talking about like the freezing level, not up to the 20,000 feet. Yeah.
And Nicola says, "Yeah, imagine the
Yeah.
UV index. Right. That's huge. Yeah.
I agree. Yeah. You've got those big leaf ears. Like it just looks like, formula for sunburn and potential disaster. All kinds of challenges up there, the whole range of them. Yeah.
So Barb Max says there must be insects in the soil. Extreme temperatures would cause weathering of the rocks. Would it be interesting to know what the lava is composed of? Would that indicate soil? Hmm.
Yeah. Great. This is exactly why I really like studies like this. It just piques your curiosity. And I think that actually the authors do a good job of kind of inspiring, right? So as you pointed out, they bring Hemingway into the conversation and that frozen
Yeah,
cougar and
yeah,
yeah. But then, that the ending by saying we have no explanation for what
yeah,
like that. It's an invitation to do exactly the kind of
yeah.
questions like Barb and Nicola and Tom are bringing to the table here. Yeah, that's exactly right. It's sort of like that, and actually, I think that's important. Actually, that we're as scientists, and that like when we give a lot of lip service to trying to broaden the conversation, engage audiences, etc. But in how we practice science, that's sort of where the rubber hits the road in a sense. And so, are we? Even the way we think about the science, are we bringing others into that process by sort of doing why we got into it in the first place? And so that's another fun part about this paper is I think it just like gets you thinking. Like you see those pictures of those volcanoes, start considering what this means,
Hmm.
and then it's like, okay, as these mice are marching up higher and higher elevations, our brains are also sort of like marching and trying to figure out what might be going on here. The more we think about that as humans, the more like a new idea might come up, or sort of like, oh, I didn't think of that. What if
Yeah.
we did this experiment? What if we did this? Yeah.
So Philip says we had domestic mice adapt to cold temp environments, cold store insulation by growing long coats of fur in the past. Musculus. That's interesting.
Yeah, absolutely. There's been all kinds of, sort of microevolutionary courses that have been done with mice and other species, and you can see a lot of that adaptability. And I think here in the Andes volcanoes, it's like, let's
Yeah.
take this to the absolute limit. Yeah.
So Jeff says, I'm driving around doing errands, and it occurred to me that it might be virus driven, like the caterpillars that seek the top of the tree.
Yeah. Now we're talking. I like that. So that's in that sentence from the authors too, right? Like, why are they seeking those altitudes? And so, certainly the mice aren't getting together on the weekend and saying, oh, well, if we went about 500 meters higher, that means we wouldn't have to worry about these other mice or these other predators. Like, that's not what's happening here. And so, yeah, bringing the infectious microbes into the conversation, that are, this idea that they're using their hosts as vehicles, as vectors to transport themselves, is pretty interesting. And so in this case, I think if the analogy holds to the caterpillars at the top of trees, does getting at higher elevation in a volcano, does that contribute to the broadcast then so as the mouse mummifies, if we hypothesize, as Jeff is doing here, a virus situation, does that mean that the virus gets farther? In which case, now again, naturally, the so-called extended phenotype, natural selection can act on that, because then the viruses that are changing that behavior to cause them to seek those higher altitudes, if those are the ones that survive because they broadcast farther, now you have that sort of renewing cycle that natural selection, those are the ideas still being studied in real time.
Barb Mack says you'd need mouse cam up there.
That's right.
Maybe Ayacuca would go for mouse cam. I don't know, right?
Yeah, that's right. Well, you don't have the bears, so we solved that problem. You don't have to sacrifice grad students in tents to the bears. You can but yeah, you want to keep an eye on them. That's right. And provide them some resources, but not too many where it becomes too cushy. Yeah.
So 3D wants to know, can we do a session on how manatees in Florida are evolving? Is it due to climate change or motorized boats or change in their food supply? Didn't we have someone once that was studying manatees?
Yeah, so this is my colleague just across the hallway here, Nathan Clark. And so we had him on for a different study on hair, evolution of hair and mammal. But we didn't have him on, but we did his paper from a few years ago, which is where he's looking for coevolutionary signals, in this case of gene loss. And so what he lifted up there was that the manatees have lost this enzyme that might relate to something to do with their metabolism or physiology with living in the water as a mammal that's returned to the water. But he now has evidence of this in beavers and in whales, other species. And the implication is that the loss of that gene, for whatever reason, and the selection might have acted to take that gene out of the population, leaves the manatees potentially more vulnerable to the pesticides that are used in the farmland that's basically contiguous to the kind of mangroves and that ecosystem where the manatees live. Yeah, that's a population under some real stress. A lot of it human imposed for the reasons of motorboats, food supply, habitat, but even these, and it goes deeper than that, and that from Nathan's work. So that's go back.
This is a great title. Let me show this, Nels. Because the title of this episode is just great. TWiEVO 34, you don't know what you've got till it's pawn.
There we go. And
remember that?
That's right, I do now. So this is pawn 1 is the gene. And the title here, we were the song, we were you don't know what you've got till it's gone. The Joni Mitchell, yeah. Song. So we were
Big Yellow Taxi. Yep.
There it is. Yep. So if you're interested in manatees, start there, and start with Nathan's paper. We covered that. Now that we're on to TWiEVO 90, we've got a good catalog we can draw from for, we're starting to hit the really cool sort of themes out there, and we can draw back, which is pretty fun.
Nicola wants to know if you'd need oxygen bottles to work up there. I have no idea.
Yeah, I don't either. I'm not a high elevation climber. I mean, obviously, so we as humans have figured out how to be relatively comfortable for short amounts of time. But we're not living up there either by any way, shape, or form. We're just sort of tourists. And, I mean, that getting again at that sort of why do humans seek these altitudes? Is there some like kind of innate impulse to explore? I have no idea. We're getting to sort of some complex, almost, potentially social evolution somehow as well.
So Barmack says some people can't handle that altitude without getting sick. I imagine oxygen would be needed.
Yeah,
All right.
for sure. And that's, that would be fun too. If, depending on the ability to bring these critters into the lab, what are their high elevation adaptations? This has been an active area in evolutionary biology in the last decade or so, is when you look at, so certainly not, 6,000 meters, but high elevation human population. So in and around Nepal, for example, and geneticists using association studies and other approaches, including some evolutionary approaches, have proposed modifications to our genes or mutations of our genes or adaptive changes in things like how hemoglobin is working or hypoxia, the lack of oxygen is handled by cells and things like that. And so it looks like there's multiple sort of physiologic routes, certainly for humans, that might apply to other mammals. But that's another reason I think to sort of explore this group of high elevation species. Have the mice converged on some of the same things? Or are they doing different things? Or by learning how the mice adapt, can we then look and ask how might what's our own capacity of our species to adapt to high elevation as well?
All right. So Peter wants to know, can you explain what's meant by coefficient of kinship and average genome-wide nucleotide diversity?
Yeah. So this is getting those sort of population genetic measures. So I'm not going to really be able to do justice to it, to be honest, Peter, out of the box here. But so, we're looking at this is looking at genetic relatedness and in sort of various in different sort of ways and in more sophisticated forms, the population genetics here. So the average genome-wide pairwise nucleotide diversity, this gets at, now that they've got, the set of 40 genomes, one of them at high quality, so high copy number, and then the other one's at low copy number. So the low copy number ones aren't like, you wouldn't use that to like say, okay, here's what we think the chromosomes all look like, but you would use that to say, here's what we think the mutations are in this guy compared to the other one. And so, as you can get a genome wide, so you can use the whole genome because they've done that. And so then what you're looking at is pairwise nucleotide diversity. And this is at any position compared to the next guy. So it's just a pairwise comparison. Are you the same or are you different? And then you do that across the whole genome, take an average, put this into sort of the whole thing. And then that gives you a sense of, among that sample, how differentiated are you or how similar are you based on each of those pairwise comparisons. And the answer here was that there wasn't a lot of differentiation. So like, if you're different at one pair versus another, that's pretty consistent through the population. The other one, the coefficient of kinship, right? So this gets at, again, don't worth digging into in real life than sort of depending on this sort of podcast definition here. But here now you're asking about relatedness. And so this gets at, if the kinship is high, then that says something about the sample that's been breeding. Because they're collecting from different, volcanoes in different places, the notion that they would actually recover high kinship isn't clear to me, or if they have enough samples. And I have to confess, I didn't go deep enough to really get at that. So like, if you take all of like the ones that they're sampling from one volcano, are these brother, sister, son, daughter kind of relationships, or are they just random ones? That also starts to get at like, just think about, okay, are they just climbing up the mountain in sort of individual sort of forays, or are they living there long? And again, I think the answer is not super clear, mostly probably just because the sampling is so minimal. And so the authors didn't really try to hang a strong sort of one hypothesis or another. They just sort of throw up their hands and say they have no explanation for why the animals are seeking those altitudes.
All right. Kip wants to know, aren't there any hot springs up on the volcano? When we climb up Mount Shasta, there are these springs with warm water all year long.
Yeah, good question. I don't know. Yeah, it'll depend on the activeness of the volcano. Excuse me,
Yeah.
sort of the geography there, whether there's, and then if snow's melting or it's pooling. If it,
Yeah.
depending on the volcanic activity, does that bring another sort of wild card, which are like noxious gases into the mix? So
Yeah.
yeah, they're pretty interesting questions. Yeah.
So Tom says bleak looking landscapes can harbor a lot of non obvious to us life.
Yeah.
Intensive ecological extant population survey would reveal a lot, including food sources. One live mouse, likely more are up there. Yeah.
Yeah, I agree. Yeah, yeah. And even 40 live ones, I guess, across the 3 volcanoes. And I didn't take a deep dive either into the sort of the extent of the sampling. I think they note that it's over 2 years, but a lot of those details aren't sort of sketched out. But it's a really good point where um, maybe as a corollary to, like, most of evolutionary biology are the stuff you can't see. Most of just like living on Earth in
Mm,
environments like right, a
yeah.
pretty there's a pretty strong imperative to, like, not just be hanging out obvious for potential predators to come in and scoop you up. So even in less extreme environments, a lot is
Yeah.
hiding right under your eyes, right under your nose.
So Barbmax says it'd be interesting to know the red blood cell count of these mice. They must need a lot
Yeah,
for that out. Yeah, that's another thing you could look at, right?
I agree. So I think the best comparison would be like the closest related species,
Yeah.
and it might be the same species or the closely related population that has lived in confirmed extended amounts of time at high elevation, at low elevation, and then
Yeah.
start to put them through the paces of, like, what's your physiologic response. It's like for humans, in some of these high elevation populations and low elevation populations, the researchers will put them on treadmills and then take all of these kind of physiologic readings, to get a sense of how they're, as you're jogging along or exerting yourself, how you're responding, and then taking blood samples and comparing some of the blood cell chemistry or counts,
Mm.
chemistries, all of those other things. And so, yeah, you can imagine of course you could even do more of that with the mice if you have good collections. Yep, totally cool. And then ultimately, where I get excited as a geneticist is, then can you link that to genetic changes to sort
Sure.
of get at the
Yeah.
basis of how these things are unfolding as well. Yep. Good points, Barb.
So Barb says 8,000 meters is the death zone. So mice surviving at 6,000 is pretty amazing.
Yeah, I think for me, like, the death zone is 10,000. Like, if I'm at a meeting up at Telluride or something at 8,000 feet, I'm pretty miserable. So not
Yeah, I
not the death
get
zone,
headache.
but yeah,
I get
same here.
my heart beats fast, and I
Yeah,
have headaches for a
yeah,
day or two. Yeah, yeah, it takes some
exactly. Yeah.
acclimation. Rob says you don't know what you've got till it's a pun. There you go.
Uh-oh.
And Priya, thank you for your contribution to science communication. I haven't seen you for a while. I've used to see you a lot on the live stream. Good to see you again.
Yeah, thanks, Priya. That's great.
The topic
Appreciate
of altitude sickness was on this podcast. Will kill you. I
that.
haven't seen that. I haven't
Hmm.
listened to many of those, but they cover things that could kill you. Yeah.
Yeah, very
So
cool. Yeah, thanks for the tip. That's like on that's sort of what we cover too on This Week in Evolution, are things that could kill you. That's a pretty strong selective pressure
Nicola
for
says, I went
reproduction.
on the Alps as a kid, I think
Yeah.
3,000 meters max, but I remember
Yep.
being super tired after a little walk. That's when I learned that my natural habitat is sea level, asphalt, and concrete.
There you go.
And Carol is making a trip to Bhutan next year. I live at sea level. I'm going to have to train to handle the altitude.
Great idea.
And
That's what those Andean leaf mice are doing. They're in training.
3D says, when I return to New York after a Colorado trip, I can feel the richness of oxygen at sea level.
Yep.
Or Max says, when people go to climb Everest, they have to go up and down between the base stations repeatedly in order to accustom themselves to the altitude. So it can take a few weeks to get to the summit.
Yep. That's right.
Have you ever climbed Everest, Nels?
No, I've been pretty modest in my mountain climbing attempts. So I think like 3,000 meters, but not climbing necessarily. So the closest I get, like, on a routine basis here in Salt Lake City in the Western Rocky Mountains, is skiing. And we get up to 10,000, 11,000, 12,000 feet. But that's like, you ride a chairlift up, and then you're up there at the top. And it's usually the wind is whistling, the snow is up there, but you're down in like 5 minutes, enjoying like lunch and coffee and beers.
Yeah.
And so it's a whole different lifestyle from what we're talking about, like living in these extreme environments versus visiting or recreating there, for sure.
Priya says, when we go to high altitude, we take acetylzalide for 3 days. You could do that for going to Bhutan. All right. All right. Peter
Very
got
cool.
altitude
Nice.
sickness
Yep.
once after driving from Austin to Breckenridge. Yep.
Yeah.
It's not fun. Altitude sickness is not fun.
Yep.
All right. Thanks for the comments, folks.
Yeah, that was great. Okay, speaking of comments, should we hit the mailbag here? We've got one email I'd like to
Yeah,
cover.
we do. Why don't you take that, Nels?
Sure, be happy to. So this is from Fernando. Hi, Nels and Vincent. I just skimmed this. It's a link to a paper here. And the title is a single cryptomonad cell harbors a complex community of organelles, bacteria, a phage, and selfish elements. This was published, I think, a month or so ago in Current Biology. I hadn't seen this yet. Thank you, Fernando, for bringing this to our attention. And Fernando notes, pretty wild symbiosis evolutionary conflict story. Algae, bacteria, virus, transposons, all thrown together in an uneasy alliance. The lead author is at UCSD. It would be amazing to hear her story. This project must have had many surprises. I couldn't agree more. This is great. And cool. as I was hinting at maybe in the opening, just in the last month or so, that I feel like cool papers in evolution kind of go in these bursts, and we've had one of these bursts here. This is one of them. And so the group here, the lead author is Patrick Keeling. He's located at UBC, the University of British Columbia. And the first author that Fernando is referring to, this is Emma George. I think she just got her PhD somewhat recently from the Keeling lab and is now at Scripps in San Diego. I'm not sure at what lab. That's a great suggestion. It'd be really fun to hear Emma tell us this story. Just from a quick glance at the paper, we've been talking about complexity in extreme environments, but what if the cell itself is sort of an extreme environment or a complicated environment? And so these cryptomonads, these algae that have plastids here, they did sort of a deep dive sort of within the cell or within the genetic ecosystem. And so you've got 7 different genomes housed by this single cell. There's the nucleus and the, sort of 30,000 genes there. But then there's the plastid that has a few hundred. There are these symbionts that bring 1,000 or more genes. And then the viruses, the phage, that bring another 50 genes to the mix. And so you have these sort of conglomerations. And by the way, of genomes, so to Fernando's point, the potential for conflict here, right? So each of these compartments that has genes, you can make the case that selection will be acting a little bit differently to the advantage of that compartment, sometimes even to the detriment of the whole cell. And so how you hold all of this together from an evolutionary standpoint is pretty wild. And then also kind of with that whole kind of fleas of fleas energy where the algal species is housing a symbiont, the bacteria, which is then, infected by the phage. And so you're just sort of marching down the layers of organization there as well. It's really cool. Thanks for sending that.
So Priya's at, it's 1:30 in the morning in New Delhi. Stayed up after a dinner party just to watch your chats. Die-hard fan from India. Yeah, I recognize your name from previous live streams. Priya, haven't seen you in a while because the hour is, they are 8 p.m. Forget it, right?
Yeah. Thank you for staying up.
Thank you for stopping by.
Yeah, that's really cool.
And Noir points out, so Breckenridge is very high, and Santa Fe is pretty high too, 7,000 feet.
Yep.
Right.
No, it hits you. It matters, which is like with that even hint, now, add another 10,000 feet or 13,000 feet to the mix. And that's an extreme environment. So Vincent, I won't go through all this, but hard to pick a single paper today. I've got another 6 here or so that caught my eye in the last week or two. There's more really cool, really interesting cancer evolution. So there's a recent one in Science about these transmissible cancers in Tasmanian devils. Right. And they do a comparison between the two. There's one that just dropped yesterday on bowhead whales and exploring Peto's paradox. This is how can these long-lived, massive mammals with, orders of magnitude more cells, for example, than we have, how do they not just croak from cancer, let alone like in our own lifetimes or early in lifetimes, but then living, for 200 years plus. And some interesting new work that ties that to DNA repair, and that repair is more important than sort of replacement of cells. And so that's an interesting new one. There's some great recent virology out there. Cheating leads to the evolution of multipartate viruses, maybe a little echo of Fernando's suggestion. And then also some great stuff on multicellularity using yeast, and then some cell biology ideas about how the size of the nucleus to the cytoplasm might influence whether something can make that massive evolutionary transition from single cell to multi-cell. One of,
Oh, so I know this de novo evolution in yeast. It's Bill Ratcliffe, who's at
yeah,
Georgia Tech.
yeah.
And
Did you meet him recently?
I met him when I was there in 2019, I think it was. And I said, we have to get you on TWiEVO because this is cool stuff. And now I see that he's published it. So maybe that's a jog to get in touch with him and get him on. Yeah.
Let's do that. Yeah, that'd be fun. So yeah, this is the snowflake yeast system. And maybe we'll just leave that hint now and see if we can get Will Radcliffe over for an interview.
So now, is cancer contagious in Tasmanian devils?
Yeah, that's right. And so that's what this other paper that just dropped in Science takes samples from two of these. It's not just a single transmissible cancer; it's several. And then sequences. I haven't done a proper dive into that one yet, but sequences them and compares them to kind of. It's almost like there's sort of different levels of conflict or competition here. So obviously the Tasmanian devils are getting the worst of it, but the cancers themselves are potentially vying for different hosts as they're transmitted. And this is generally through fighting. And so you get these like really disfiguring facial scars on some of these Tasmanian devils. This is a host population that's already threatened anyway. And I think the most recent idea is that there's actually some resistance that's coming up in that population. And so if the humans can get out of the way and sort of support that species and limit habitat loss and things like that, then likely that they naturally weather the storm of these transmissible tumors as more and more resistance builds up in the population. But yeah, a really fascinating system there as well with some of that complexity cooked into it.
All right, one more from Carol. I heard that having short telomeres doesn't necessarily shorten your life.
Yeah, that's right. This is another one that ripped from the headlines of Science in the last week or so. Yeah, I saw that report too. I haven't had the chance to do a deeper dive there, but it's really true. I mean, we see this over and over again, which is sort of the when we think about complex biology, like in this case aging, and then you find markers or correlates of that, which can be really intuitively attractive, right? So if you have telomeres. As most of you already know, this is the ends of our chromosomes, and you have this problem when your genomes are comprised of linear chromosomes, is that when you're copying them as you're dividing a new cell or reproducing, there's an erosion in that replication process at the end of the chromosome, and so the telomeres solve that. And so the notion would be, and with some evidence, by the way, over the years, held up that the longer the telomere, the safer the chromosome is, the younger, quote unquote, the chromosome is, or and so maybe that would influence lifespan. That's certainly 100% true. Like if you're a budding yeast cell, for example, but if you're a human or a mammal, the story is a little bit more complicated. And so for a lot of this complicated biology, the rules that we might learn or the patterns or evidence that we gain in model systems might not always translate one-to-one. And so that's another example of like, as you're doing research, like how are you weighing what you're learning and what are the compromises you're making to like understand the rules by which sort of things work. But that doesn't necessarily apply one-to-one, the fundamental science to the sort of translational as we all strive, to live to be 200 years old, like a bowhead whale.
Hello, Sundari Ann from South India, 1:46 a.m. Just found this. Glad you found it. Unfortunately,
Wonderful.
We already did the main paper, but maybe you want to go back and listen to the recording.
There you go. Rewind the tape on YouTube. It's right there, and good to be together, even if it's just for a few minutes.
Right. All right,
Very
Nels.
cool.
Want to do some picks?
Yeah, let's go to our science picks of the week. So, speaking of more science emerging and more publications, so my pick of the week is this effort called Zoonomia. And got the link here. This is an international collaboration to discover the genomic basis of shared and specialized traits in mammals. So kind of building off of, the a very specialized mammal that we just talked about in terms of being able to tolerate high elevations, the Andean long-eared mouse. But Zoonomia is sort of a genome project for mammals, centered around that idea that by comparing the genomes that we can start to, see what's common among us and what's different. And so now, and just published, I think, made a nice splash the last week in one of the glossy science magazines, to report sort of the first wave here, which I think is about 300 species of mammals with high quality genomes. And the key here, to I think point out or pause and consider, is high quality genomes. So there have been a number of efforts to just say we sequence the genome of XYZ, or there's been 100 genome project, 1,000 genome project, 1,000 mammals, all this stuff. In many cases, that's sort of can become these like flag planting exercises by scientists who are just trying to like show off a little bit that they can sequence a lot of stuff. The hard work is to, and it's becoming like the front end is becoming a little bit easier as the technology gets better. And we saw that for the Andean leaf-eared mouse, where there's now a 30x genome. But the really hard work here is the annotation. And what I mean by that is to take all of the raw data and then to understand what are the parts of the genome that are coding for genes. And, is that like high quality, and how many gaps are there? How much, sort of the nuts and bolts of what's really inside that genome. And is there a way to search that? What Zoonomia does is it makes it a little bit easier to do that. We've been messing with it before the kind of publication with bats. There's something like 50 or 100 bats in there. And so if we're interested in a gene, we can now go to their website, go to their database, and because the annotations are decent, we can go in and pull out the gene we're interested in and compare that across, like 50 comparisons. Still not perfect. So like if you are getting to that high of a number, like if we pick any one gene, it might be missing from 10 or a dozen of them, or the sequence at that location might be low quality. And so when you go from that sort of idea of we've sequenced a genome to actually working with one day to day, those are two very different things. And so this is a real step forward. And already, in this sort of this international collaboration, they're starting to take, some really nice examples now that they're starting to invest the resources to get these good genomes. What can you learn now? And it's sort of rewriting the textbooks in terms of how are mammals related to each other? When did they specialize in different ways? And so really cool website too, that we'll put on the science links here in our picks, or you can just do a Google search for Zoonomia, Zoonomia, and start to explore kind of at all levels this rich data set.
Cool. That's very nice.
Yeah.
It's a nice website. Very good.
Yeah. How about you, Vincent? What's your science pick?
So I want to pick one that's probably been picked before on TWiEVO, but this is a good one. It's called C-phages.
I'm familiar. This is awesome.
This is a site. Well, it's a program. It's a program of HHMI and C-phages themselves. They've merged, but it is Science Education Alliance, Phage Hunters Advancing Genomics and Evolutionary Sciences. It's a 2-semester undergraduate research course where students discover their own phages. You dig in the soil, and then you use it to infect a mycobacterium. You isolate phages. You can do genome sequences. You can annotate genes. You could name the phage after yourself, and it just teaches a lot of independent discovery here. And the website is very nice. They have a lot of information about what's going on. This was the brainchild of Graham Hatfull at the University of Pittsburgh, who first thought it up many years ago. And I have to tell you that when you search for TWiEVO Hatfull, here, let's do that. You come up with TWiEVO 87, Nels.
All right, wow.
Fireside chat with Professor Graham Hatfull. Look at how young he is there.
2010, about 13 years, almost exactly 13 years now.
This is when we just had audio. We didn't have any video. He established this, and now it's a very big system that is deployed all around the world. A lot of people helping them out, and you can see here that you can find various phages. So they have genome exchange, phages database and so forth. So if you want to have this at your high school, you can go to that link and figure out how to do it. It's
Yeah,
really cool.
it's really incredible. That would be another potential guest, maybe, 15 years, 13, 15 years on, update from Graham about the success of that program.
Yeah.
It's really incredible. I've been inspired by that. Easier said than done, but could there be a C phages-like approach? Instead of discovering the viruses of bacteria, could we do that to discover the viruses of zebrafish?
You could.
C phages in the Z fish or something. Yeah, that's kind of a little idea percolating. But yeah, have you talked to Graham since you had him on TWiEVO? I met him a few years ago.
I have met him now and then, but I haven't.
Yeah.
He hasn't been back. So
Yeah. Maybe it might be time for TWiEVO, because evolution is right in that.
for
Sort
sure.
of the ideas there.
And we could get him on a live stream. That's fun. I like
I do
having
too.
guests
Yeah.
on live streams. It's just a different way to do it, and you have the live questions, which I think are a lot of fun. So
Really fun. Yeah, and today's episode was a perfect example of that. Thanks to everyone for tuning in from around the world and bringing these questions, that conversation. That was really fun.
yeah, so thanks everybody from coming. We had for coming. We had 67 people at the moment, and we have 65 likes. So that's great. Thank you so much.
Appreciate it.
And thank you to our moderators. Today we had Steph, we had Tom, we had Barb Mack,
Barb. Yep.
we had Tom, right?
Mm-hmm.
I think I got everyone, right? Did I miss anyone? We had Les, right? I forgot Les. There you go.
Yep.
Thank you all for moderating. And don't forget, if you're awake tonight at 8 p.m. Eastern, we will have office hours. My guest will be Dan Wilson from Debunk the Funk, and he will come on around 8:30. I'll start at 8
Cool.
and field questions, but he has to take care of his daughter, his young daughter, and put her to bed, and then he'll come. Nels, thank you for doing this again. It's always a lot of fun.
Yeah. Wow. 90 in the books, Vincent, and 91 coming.
So
In a few weeks.
90, so 10 more. That'll bring us to next year, right?
Yeah. Yep.
So
Kind of early.
for 100, we need to do something different, right?
That's right. We'll do something in vivo for sure. And I've got a feeler out right now on a possible venue in the hill country of Texas. I'll report back on that one next time.
All right. That's great.
Yeah.
All right, everybody. Until next time, we'll be back in about a month. Do we have a date for the next one yet, Nels? I don't remember.
We'll probably not yet. We'll do that offline
All right.
after we wrap up here. Yeah.
All right, Tarol, thank you for your contribution to science education. I'm trying to highlight it here. There you go. Thank you for
There
your
we go.
contribution.
Awesome.
Really appreciate it. Thank you, everybody. Be safe. See you next time. Bye-bye.